We will wait probably another 1 or 2 minutes to get a few more people join us. Thanks for your patience. And I apologize as well to the people that did attend the other sessions. There will be some repetition in the beginning, but rest assured, the presentation itself will be definitely different and a great one. So I guess I'll start. So yeah, good morning, good afternoon, good evening. Welcome to the session 5 and last of the Waters webinar week, focusing on innovations and applications for the clinical workflow. So I'm Golo Bosch and I'll be your host. And before we get into the presentation, I'd like to share a few practical details. At the bottom of the screen are some materials. They are different for every session, so these are relevant to this presentation. There might be some that are repeating from one session to another, but some are really very specific to this one. And then you have a Q&A box that, again, we'd like you to use as much as possible for the questions you have about the presentation. And we'll answer as best we can at the end of the presentation. And then we also would like you to fill the survey, which is on the right of your screen. And that allows you to give us feedback and tell us how we did, but also indicate probably some future topics that we could address and allow us as well to improve. So today's presenter is Rob Wardle. He is currently a senior scientist working at Waters Mass Spectrometry headquarters in Wilmslow in the UK. And that is in the clinical business unit created 18 months ago by Waters Corporation. Rob joined Waters in 2010, playing a key role in reagent kit verification activities before moving on to developing LC-MS methods for a range of applications, including vitamin D metabolites, estrogens, and a large panel of steroid hormones, where he also included a complete LC-MS workflow of these applications areas through offline automation and LIMS integration. And yes, Rob is definitely an automation specialist for us. His presentation title is Analysis of Serum Estrogens for Clinical Research, and the show is yours, Rob.
Great, thank you very much, Godo, for the introduction, and welcome everybody. So within this presentation, I'll briefly describe the steroid hormone pathway and some background into estrogens. Then we'll move on to some of the challenges faced, such as the isotopic interference challenges, and also discuss the importance of chromatography to separate these out. I'll also look at how we deal with endogenous type of compounds, such as the estrogens, and then we'll take a look at our solution and how we have tried to tackle these challenges. And I'll also show some of the method performance data. So natural steroid hormones are all derived from cholesterol through two main functional classes of enzymes to form the steroidogenesis pathway. And these steroid hormones play many critical roles in the body, which include regulating water and salt balance, metabolism and stress response, and also initiating and maintaining sexual differentiation and reproduction. And the estrogens form a part of the steroidogenesis pathway and they're shown in the bottom right-hand section of this image. So estradiol is produced primarily in the ovaries and testes by the aromatization of testosterone, with small amounts also being produced in the adrenal glands and some peripheral tissues such as fat. Estrone, on the other hand, is primarily produced in the adrenal glands by the aromatization of androstenedione, and both estradiol and estrone can also be converted to one another through the 17-beta dehydrogenase or aldoketoreductase enzymes. And in pregnant women, both estradiol and estrone also can go on to produce estriol, or E3, in the placenta and liver. And the role of estriol is still not fully understood, but it is known to have a much lower affinity for the alpha and beta estrogen receptors compared to estradiol and estrone, and this makes it less biologically active. Estradiol circulates in the human body at higher amounts when compared to estrone in premenopausal non-pregnant women, and circulates at much lower levels in men and postmenopausal women. Estrone, on the other hand, actually differs less, and this results in a reversal of the estradiol to estrone ratio where estradiol levels are lower. And this can be useful in investigating certain conditions. Estradiol's main role in females is maturing and maintaining the reproductive system, but also has additional roles in both males and females, such as the way fat is distributed and bone maintenance. So now I'm going to go slightly off-piste, as I am a keen cyclist, and I often encounter many challenges when riding my bike, especially on UK roads. And you can see some great examples in the images shown here. So things such as potholes, flooded roads, or well-positioned obstacles in cycle lanes are some of the things that regularly encounter on a bike. But what if these challenges became more like the challenge that is analysing estrogens by LCMS? Well, they would probably look a bit more like this, where potholes would turn into complete roads disappearing in front of you. Puddles may turn into a whole new balancing game, trying to stay dry, and a simple obstacle in the road may turn into a complete slalom course. So why is analysing estrogens so challenging? Well, the main challenge really is that they're required to be measured at very low concentration levels. And this is ideally at around 1 picogram per mil or 3.7 picomoles per litre. And as they're non-polar molecules, this results in them having poor ionization efficiencies in the mass spectrometer and limits what sample extraction techniques are also can be used. Also, the fact that the estrogens are endogenous, this presents challenges analytically. Things like obtaining a suitable blank matrix, and also performing the required method verification tests, such as the matrix effect tests. And another consideration is that their molecular weights only differ by 2 Daltons, where estradiol has a hydroxyl group on the 17th carbon, whereas estrone has a double bond oxygen. And this means that because they're so close together that they require separating chromatographically to prevent estrone from interfering with estradiol in the mass spectrometer. So now I just wanted to take a look at each of these challenges in a bit more detail and just discuss the approach that we took with our method to try and overcome these, and hopefully show you that actually it's not as scary as maybe my previous slides suggested. And also how the advances in the technology as time's gone on can help to overcome these challenges. So the first challenge is to overcome the poor ionization efficiencies to improve the overall analytical sensitivity. And one way to improve poor ionization is to derivatize the sample. However, this is seen to be time-consuming, a challenging process to undertake, and also it can be costly, and so really it's not that desirable to perform. Therefore, during our method development, we investigated the use of different mobile phase additives in an attempt to improve the overall signal-to-noise ratios. And this testing revealed that using a very small concentration of ammonium fluoride in the mobile phase gave a large improvement in ionization efficiency when compared to simply using more conventional additives. Excuse me. And this figure nicely highlights these improvements. So you can see here how our blue chromatogram containing the ammonium fluoride in the mobile phase is significantly has a greater response when compared to the red chromatogram. So the second challenge I was going to discuss is how we evaluate matrix effects when dealing with endogenous compounds. So firstly, we'll look at how the test is typically performed, at least in our lab, and then show how we've overcome the challenge of estradiol and estrone being endogenous to perform these tests. So the matrix effect evaluation are described really well in the CLSI C62A edition 2 guidelines, which references a great paper by Matuszewski et al. And it describes the best practices of evaluating at least 5 different native matrix samples to perform a quantitative test to evaluate matrix effects with total allowable error limits, and also ensure that the sample matrix affects the analyte and internal standard to the same extent. So a good way to estimate the impact of the presence of the matrix on analysis by LC-MS during our method development stage is to actually perform a qualitative test. And this schematic on the slide nicely shows this process. So what we do is we extract a matrix sample and inject this onto the LC-MS system. And this is done without actually adding our internal standard. Rather than inject it in the normal way, however, after the LC column, we will infuse a standard solution of the analytes and internal standards through a T-piece into the LC flow, which then goes into the mass spectrometer. And what this does is that rather have a background being quite low, it will actually raise this background up because you have that constant stream of your analytes being detected. And this then gives the ability to look for these suppression and enhancement effects within the injection of the matrix, where suppression effects will be seen as a drop in the baseline and enhancement and an increase in the baseline. And these effects are common, even with really clean sample extraction methods. However, what we're looking for with this test is that these suppression or enhancement effects don't occur at the same retention time as our analyte peaks when performing a normal injection. So the next couple of slides show examples of this. So this is a good example of where iron suppression may cause a problem with a method. So you can see that the green trace is showing lots of suppression areas, and also where we have a gradual increase in our response over time, possibly due to a large suppression effect, which gradually improves over time. And then here we actually show an example of the qualitative matrix effect test for estradiol. So you can see here the blue trace is actually a solvent blank. So rather than injecting an extracted matrix sample, it shows just an a solvent injection, and this gives a nice baseline to work from. The red chromatogram, then shows our injected blank matrix sample, and the green peak is our retention time for estradiol. And you can see that the circled sections with the orange circles are our suppression areas, and all of these are separated out from our peak, and so we deem this to be acceptable to move on to the next stages of our method development. And then to perform a quantitative matrix effect test, we extract blank samples with no analyte or internal standard added. The final extracts are then spiked, post-spiked with internal standard and low concentrations or high concentrations of our analytes. And typically, we would perform this experiment with triplicate replicates of each of our low and high concentrations, and in each of 6 individual matrix samples. We would then also spike a solvent solution in exactly the same way, and all the samples would be injected onto the LCMS system. The impact of the matrix, or what we call the matrix factor, can then be calculated by taking the mean peak areas of the analytes in the presence of the matrix, and then dividing those by the peak areas of the samples with no matrix. And then any values that come back to be less than 1 would indicate that there is some matrix suppression, and a value of greater than 1 would indicate that there is ion enhancement. However, this approach obviously doesn't take into account our endogenous contribution. And so how do we account for this? So what we do is we take exactly the same approach. However, the response in the presence of the matrix is done by performing additional extracts of the matrix from each of the 6 individuals without any post-spiking of our analytes. And then the mean peak areas observed from the matrix-only samples are then just simply taken away from those with the post-spiking. And this will give us the response of that post-spike in the presence of the matrix, excluding those endogenous levels. So here we have an example of one of our individuals for estradiol. And as you can see from the right-hand column in the top table, we did actually see some enhancement for our mean peak areas with the calculated matrix factors. However, what is also important to note is that this isn't necessarily a problem if the internal standard is affected in the same way. So therefore, it's also important to repeat the calculations, but taking into account the analyte to internal standard ratios. And as you can see in the bottom right-hand part of the the bottom table on the right-hand side, the internal standard actually compensates nicely for that small enhancement that we've seen. And the acceptance criteria that we apply on this test is plus or minus 15%. So they come well within that. And the final consideration with such compounds is how do we select a good surrogate matrix? So we opted to use a stripped serum provided by Golden West Biologicals, where we found their MSG 4000 product to be a very good surrogate matrix for analyzing estrogens. And this is certified for the steroid hormones. And as you can see from our blank extract, the example shown, we saw little to no observable peaks for estrone or estradiol within this matrix. And the final challenge I wanted to discuss before I come on to our solution was the isotopic interferences. So firstly, what is an isotope? Well, this is defined as two or more forms of the same element that contain an equal number of protons, but a different number of neutrons. And this will result in it having a different atomic mass, but the same chemical properties. So some naturally occurring molecules contain a small percentage of isotopes. And if we take carbon as an example, this has a mass of 12 Daltons, but approximately 1% of all naturally occurring carbon exists with one extra neutron, known as carbon-13. And this has a mass of 13 Daltons. As estrone, for example, has 18 carbons, this will result in 18% of all estrone existing with one carbon-13 within its structure, and therefore it would have a molecular weight of 271.4 Daltons, and this is 1 Dalton higher than the main peaks. And there's also a 4% chance of estrone having 2 carbon-13s within its structure, and therefore having a molecular weight of 272.4, being 2 Daltons higher. And you can see this pattern nicely in the mass spectrometer when the compound is infused. So you can see at 269.3, we have the main estrone peak, which is 1 mass unit less because we are ionizing in negative ionization mode. And then at approximately 18% of the total response of this main peak, we see a second peak having a mass to charge ratio of 1 unit higher, and then a third peak at approximately 4% of the total response being 2 mass units higher. And then if we overlay a spectra for estradiol, you can see that this third peak of estrone actually has the same mass to charge ratio as the main estradiol peak. And therefore, a high concentration of estrone could potentially significantly interfere with a low concentration of estradiol in the mass spectrometer. And this is why it is important that we need to chromatographically separate them. So now let's take a look at our application note for estradiol and estrone. So firstly, for the sample extraction, we have a relatively simple workflow. created where only 250 microliters of serum sample was required. We performed liquid-liquid extraction, and this used a mixture of ethyl acetate and hexane, and the samples were centrifuged to separate the layers efficiently. We then transferred the top layer into a clean 96 well plate. The samples were evaporated to dryness and then reconstituted in methanol and water before injection on the LCMS system. Um, 20 microliters of the extracted samples were injected onto the I-class FL system, and samples were separated using a Cortex phenol column with dimensions of 2.1 by 50 millimeters, and this was heated to 50 degrees Celsius. Mobile phase A consisted of LCMS grade water with ammonium fluoride, and mobile phase B was just methanol. The chromatographic separation was performed across 3.5 minutes, and then the column was washed and re-equilibrated, and this gave an injection to injection time of around 5.5 minutes. And then if we take a look at the Xevo TQ Absolute conditions, the detection was performed in negative ionization mode, and the voltages and temperatures are shown in the grey box. The instrument was set to unit resolution, which is around 0.7 Daltons at full width, half maximum height. And the mass to charge ratios of the estrogens can be seen in the table for the precursor and product ions, and their optimized cone and collision voltages are also shown. So chromatographic separation, as I've said, was performed using the Cortex phenol column, and the example chromatogram shown here is of an EQA sample, which we calculated at 21 picograms per mil for estradiol and 34 picograms per mil for estrone. And as you can see, importantly, estradiol is clearly separated chromatographically from estrone, and therefore this would ensure that no isotopic interference would occur in the estradiol quantification from estrone. And just to clarify, because it's not necessarily that clear on the image, the estradiol is the first eluting peak and the estrone is the second eluting peak there. Our calibration curves were all shown to be linear over the calibration ranges described, and they had correlation coefficients of greater than 0.99 and percent bias of within plus or minus 15% or plus or minus 20% for our calibrator 1 for estradiol and estrone, and this was across 8 different occasions. We assessed our precision by extracting 5 replicates of the QC low, mid, and high samples, and this was done on each of 5 different occasions, giving us a total of 25 extracted samples at each of the concentrations. The QC samples were prepared by supplementing estradiol and estrone into the Golden West Biologicals MSG 4000 serum, and the concentrations that we tested can be seen in the tables, but it ranged from 3 picograms per mil in our low up to 700 picograms per mil in our high sample. We then performed statistical analysis to calculate the repeatability and total precision, and these results were found to be less than or equal to 4.5% for estradiol or 4.8% for estrone. Excuse me. Then if we take a look at our analytical sensitivity testing, so this was performed by extracting and analysing 10 replicates of low-level samples across each of 4 occasions. And again, samples were prepared by supplementing our analytes into the Golden West Biologicals serum prior to extraction. So we set our lower limit of quantification at the concentration where the CV was less than 20%, and also the signal to noise ratio by peak to peak was greater than 10:1. And as you can see from the table, the percent CVs and percent difference from the nominal values for estradiol and estrone are shown, and both analytes achieved percent CVs of less than or equal to 20% at 1 picogram per mil. And when we take a look at the low-level chromatograms, a signal-to-noise ratio of greater than 10:1 was not quite achieved at 1 picogram per mil for estradiol. And therefore, we set our LLOQ at 3 picograms per mil, even though 2 picograms per mil did give us a signal-to-noise of greater than 10:1 in the majority of those samples. We decided to err on the side of caution and set this at 3 picograms per mil for our application. For estrone, on the other hand, we achieved a signal to noise of greater than 10:1 at our 1 picogram per mil level. And therefore, for estrone, we are able to set our LLOQ at 1 picogram per mil. And then finally, we performed a method comparison to the UK NEQAS mass spectrometry laboratory trim mean values. And this was performed for estradiol on 40 different samples. We then performed statistical analysis, and our Deming regression analysis demonstrated a really good agreement where we had a Deming fit value of y equals 0.971x plus 4.8, and a Bland-Altman analysis showing a minimal bias of minus 4.1% when comparing to these mass spectrometry laboratory trim means. And so finally, to summarize, an LCMS method was for the analysis of estradiol and estrone in human serum was developed using the TQ Absolute IVD mass spectrometer. I've gone through the challenges we faced with analyzing these compounds, which included the low analytical sensitivity requirements, how we deal with endogenous compounds, and also the importance of our chromatographic separation for these. And then we've also shown that the method has excellent precision, good agreement to the UK NEQAS scheme, and excellent analytical sensitivity required for this clinical research method. And so with that, I'd just like to thank you all for your attention, and we'll welcome any questions. Thanks.
Thank you, Rob. Nice presentation. And definitely, I mean, you were capable of overcoming these great challenges that you have shown us at the beginning. So I see that we have a few questions coming in. So again, you still have time to type into the Q&A box in order to get answers from Rob. So let's start with the first one. Maybe an easy one. Why did you select a Cortex phenyl column?
Yeah, so I think as the slide showing the separation, the chromatographic separation showing, the phenol chemistry actually separates the two compounds out really nicely, being they both have ring structures within them, so that phenol chemistry works particularly well in that case. The reason for the cortex was the solid core particles actually gave really nice sharp peak shape, which when you're trying to improve your signal to noise as much as possible, that really helped. And the reason we selected the 50 millimeter was to ensure that we still achieved a reasonable runtime, but obviously importantly maintaining that good separation of those two peaks in particular.
Makes a lot of sense. Next one, what is your view on analyte enrichment to overcome limited sensitivity? For example, to measure estradiol on a TQS micro, and what would your approach be?
So analyte enrichment, well, it's not something we significantly looked into. I guess it depends firstly on the laboratory requirements, why they want to analyze these compounds and what specific concentration levels are required. If the more laborious technique, the more challenging extraction techniques are worth the pain, shall we say, to get down to the lower levels, maybe on a TQS micro, which isn't as analytically sensitive as a TQ absolute. If that is preferred, then unfortunately, as I say, it's not something we've looked into. We have looked into the micro for these compounds, and we actually did achieve very good analytical sensitivity on that system. However, it obviously wasn't as low as you could get on the absolute. In terms of an approach, the only thing I've really briefly considered outside of using the mobile phase additives was the derivatization, whereby I guess stanzyl chloride would be the first compound I would look at. Outside of that, there's not really much more I could say at this stage, but maybe if it's of interest, we can have a discussion offline if you want to get in touch, and we can look at this further. Certainly look into this further.
Thank you. Yeah, I think it's pretty clear. Maybe I can add as well that, well, of course, if you have absolute, well, that's probably the way to go, because if you need to do some enrichment, that means additional steps, which could result in additional variability. But of course, if you only have a TQS micro, then I understand that you would want to find a way to well reach that sensitivity. If the quantitative matrix effects test fails on peak area, is this a problem?
So if it ideally, what you would obviously want is for your peak area to be as close to 1, should we say, a matrix factor of 1 as possible, having minimal suppression or enhancement. However, in reality, that's not always the case. As I've shown in the example here earlier, I think we had some enhancement of, it was around 20%, 30% for the individual that I shown. It becomes a problem if you have a large suppression effect when you are looking at these types of analytes where you need every bit of analytical sensitivity to get down to those low levels. Obviously, if you had more suppression in those cases, you may struggle further to achieve your goals. But what is important is that the internal standard compensates for those matrix effects well, and when you perform the calculation with the internal standard, that those matrix factor values do come much closer to 1. If they don't, then that may indicate a problem, and you may suffer from different matrix effects through different individuals, which is obviously a big concern within the method.
Okay. And I think we have a last one. So you did a liquid-liquid extraction. So why did you choose hexane and ethyl acetate mixture for the estrogen method?
So again, during the method development, what we found that actually using hexane greatly reduced the amount of background interferences and also the background general noise height. However, if we extracted in 100% hexane, we also found that we didn't recover the estrogen peaks, likely because they're not soluble in the hexane. Obviously then reducing our recoveries. So what we found was when we introduced ethyl acetate into the mixture, at a certain point, we were able to get good recovery on the estrone and estradiol, but still minimizing the interferences and the background height. So there was kind of finding that optimum mixture, which really helped for this application.
Okay, it makes a lot of sense. I mean, it was the best compromise you could find to reach your goal. Excellent.
Yeah.
With this, I think that was the last question, and I haven't seen any movement in the last 9 minutes. So I guess this will be the end of the session. Thanks for all the attendees for staying with us, and thank you very much, Rob, for this great presentation. And well, looking forward to see you again in the next event. And by the way, yes, you will be able as well to watch this on demand in the future. So thanks very much. Bye-bye.
Great. Thank you, everyone. Bye.
Bye.